Good morning, student. Today we are going to learn about the different type of electrophoresis. If you look into your syllabus, you have to learn about the three different type of electrophoresis. That's one is paper electrophoresis, gel electrophoresis, and capillary electrophoresis. Here I have classified the electrophoresis first in two different categories, and then it has been further subclassified. So the first two classification for electrophoresis is zone electrophoresis, and the next is moving boundary electrophoresis. In zone electrophoresis, you have paper electrophoresis, gel electrophoresis, cellulose acetate electrophoresis, thin layer electrophoresis. Among these four, you need to study only two different type of electrophoresis. One is paper electrophoresis and other is gel electrophoresis. In moving boundary electrophoresis, here I have talked about three different type of electrophoresis, but you need to know about the only one type of electrophoresis, that is capillary electrophoresis. So let us come to the zone electrophoresis. What is zone electrophoresis? The term zone electrophoresis is applied to a system in which ionic mobility are studied on a strip of paper, cellulose acetate or acrylamide. Here you have gotten a suppression. Here you have a supporting material for the suppression of your sample. So what you do, you place your sample onto the supporting material, which is generally made up of a paper or cellulose acetate or acrylamide. So when you apply the sample into this supporting medium and you apply the electric or potential difference or electricity on this, the mobility of the ion present into your sample starts. So the migration of these anions again depends upon their charges and the size shape. So what happened after the certain time of a migration, if you will see that all this anion of a different nature is present in a form of a zone. So this is called a zone electrophoresis. That's the meaning of zone electrophoresis. So you look here into the picture. I have shown it here. I have shown you the negative electrode, positive electrode, and a buffer. I have talked about the importance of buffer in the previous class. What does a buffer do and why it is important? And then I have talked about to the application of a sample, and then to the application of application of a the application of a sample and the supporting system like a paper and cellulose and acetate. So if you look into the picture, what happened there is a migration here of your sample, and the zone is obtained. So what is the advantage of zone electrophoresis? The advantage is that it is very useful in biological investigation. Even a small quantity can be analyzed, and low cost and easy maintenance is there, because the supporting system or the system on which you carry the electrophoresis is generally a paper or a gel. So but you have gotten a disadvantage in this. So what is the disadvantage? We have learnt about the disadvantage with respect to the factor affecting the electrophoresis. So if you look into the previous class, I talked about the absorption. Where does the absorption happen? The absorption generally happen into a supporting medium where it is made up of a paper. And if we look into the other effect is in osmosis, electroendoosmosis, right? It is because of the presence of certain charge particle within the supporting medium, right? So if you look into the paper, it is generally this carboxylic acid. If you look into if it is made up of a glass, it is silyl. Or if you look into the agarose sort of medium, it is a sulfate, right? So all this minus, this all, if you look into this, they are negatively charged ions, or basically you can call it a cations, right? You can call it as anions. So these anions will prevent, or it will hinder the mobility of the negatively charged particle. And then we have talked about the adsorption. Adsorption happen because of this paper. Your sample can get absorbed on that, and the migration of this cation and ion can be affected. So it may lead into the cosmic, it may lead into no, that's called uh, that's a particular term, right? Is called, I just told you in the previous class. What was that? It was a cosmic, right, or comet effect, like you will get in the suppression, right? So suppression is not proper. So this is how the disadvantages. And the other disadvantage is molecular sieving, or which we have seen with respect to Saphadex and gel. So these all are the disadvantage. So let us now come into the paper chromatography, which is there in the syllabus. So what is a paper chromatography? You look that the supporting medium here is a paper, or generally a paper which has got a less absorbance is generally used, and is generally made up of a quality of a as made up of such a quality where there is 95% of cellulose is present. So you have to look into that. It contains a 95% of cellulose and very slight adsorption capacity. This technique is useful for the separation of even a small charged molecule such as amino acid and small protein. So when you look into this, what is there? There is a paper on which the sample is spotted, right? So I have talked about the nature of sample, what is being spotted here. So then this paper is moistened with a buffer. At the end of the strip, it is immersed into the respective electrode, and the applied sample start migrating. So the migration happen again upon the nature of the sample, how it is, and you get a separated one. So this is how paper electrophoresis is done. Next is, so let us see what is the application. It is generally used in the suppression of muscle proteins, egg white proteins, milk protein, snake, insects, venom analysis, and the other purpose is daily serum analysis can be even carried out by electrophoresis. The next one which I have shown you is a gel electrophoresis. You can see it is made up of a gel, right? The supporting medium is a gel here, and you can see the other dark picture which I have put is a separated one. You can see the separated one. So what is basically this? If you look into the separated, what happened in general gel electrophoresis is nothing other than the macromolecule like DNA, RNA, or separated protein, right? So with this, what happen? You can, in this, what we do generally, we do prepare we do dissolve a agar, and then we pour into the medium where it can solidify, and you got in a gel like supporting medium, right? It's generally made up of agar. So after this, what we do, we generally pour into the system, and then we apply the sample there, and the sample is generally DNA and RNA. So here the separation can be done for the estimation of size of DNA. It can be even done for analysis of PCR product, which is used in genetic diagnostic or genetic fingerprinting. It can be also used for suppression of restricted genomics DNA prior to southern transfer, or RNA prior to northern transfer. It can be even used in forensic molecular biology, genetics, microbiology, and biochemistry. The result interpretation in this is generally done with the help of UV light or a gel imaging device. This image is recorded with computer operated camera, and intensity of the band or the spot of interest is measured and compared against a standard or marker and loaded on the same gel. So it is a comparative study, and the measurement and analysis are done with the specified software. Depending on this type of analysis is being performed, and other techniques are often implemented in conjugation with the result of gel electrophoresis, providing a wide range of first specific application. So this has become one of the biggest separating instrument with respect to the human DNA, or the study which carries generally the biological samples. Thank you.